Hello, everyone, and welcome to our first webinar of 2023. We're just going to give it a few moments to allow everyone to hop on. Thanks for joining us. Good afternoon, everyone. Just giving it a few moments to allow everyone to hop on. Welcome to our first webinar of 2023. We'll get started in just a moment. Hello, everyone. Welcome to our first RDCA-DAP webinar of 2023. Clinical outcome assessments, does one size fit all? So we're going to go ahead and get started. I'm going to start with just a few housekeeping items. If you have any questions throughout the presentation for our speakers or about the programs in general, please put those in the Q&A box. The chat function is disabled for everyone, just so we can make sure that we don't miss any questions that come up. So if you have any questions, please put them in that Q&A box and they will be addressed. Also, just a note, all the participant lines are muted so we can reduce background noise. We are also recording this presentation, so it will be available for on-demand viewing via our YouTube channel, and that link will be shared after the presentation to everyone who registered. I've also included our general contact email address. If you have any questions or want further information after the webinar, please feel free to reach out to us there. And we'll also include the contact information of our speakers during the presentation later on. At this point, I'm pleased to introduce our speakers. Dr. Alexandre Betourné received his PhD in PharmD from the University of Toulouse in France. Before joining C-Path, he worked extensively in the rare disease space, leading a team of scientists developing therapies for ALS. As executive director of RDCA-DAP, he works closely with the rare disease community in driving projects to develop solutions in rare disease drug development. So welcome, Alex. Our second speaker, Dr. Lindsay Murray, has more than 15 years of experience in health outcomes research. Prior to joining C-Path, she worked on the exact patient-reported outcome initiatives team, eventually taking over as director to lead all analysis work. As the executive director of the Rare Disease COA Consortium, Dr. Murray directs collaboration aimed at identifying scientifically sound tools and methodologies for collecting clinically meaningful outcomes data in treatment trials for rare diseases. And with that, I'm excited to hand it over to Dr. Murray, who will kick off the presentation. Thank you all again and welcome. Lindsay, you're muted.
The mute button is getting me today, but welcome everybody. I'm delighted to be here. We have over 150 people online, so it's exciting. I wanted to just start by briefly just level setting and kind of defining the COA types. So there are four main clinical outcome assessment types. Typically, patient-reported outcome is what many people think of, where you're reporting on yourself. An observer-reported outcome measure is where, say, as a caregiver, I'm rating my child's things I can observe from my child. Clinician-reported outcome measure is something that's used where a clinician is actually watching a patient do an activity and making an assessment of their ability to do that task. And then a performance outcome measure is something like a walk test, where you're looking at how well a person is able to perform a certain task. So I kind of jumped in, but I wanted to just say we're going to give a very brief overview of kind of the COA assessment methodologies and how you pick a COA for use in a rare disease clinical disease trial, and what some of the issues related to that can be in rare disease populations in particular. I don't think it's not going to be news to anyone that there's a huge unmet burden around rare disease worldwide, and we have very few treatment options available for patients who are suffering from these conditions or living with these conditions. Tanisha, if you want to go to the next slide. So before we even really start tackling the rare problem, we have to take a step back and really think about how do we select a clinical outcome assessment tool for use in a clinical trial, and what are the pieces of information that we need to do that, make that selection. So the FDA has put together a very nice roadmap here for us, and one of the key things you have to ask yourself is starting with what do you understand about the disease? What's the natural history of the disease? What does progression look like in this disease? What does improvement look like in this disease? What are the timelines associated with that? And then you have to conceptualize clinical benefits and risks. What is your treatment going to impact? Because it may not be curative, like a gene therapy hopefully might be. You may be just tackling one symptom out of a multi-system condition. So you really need to identify what's the concept of interest. What are you trying to measure in your specific trial population? And then you have to look at what COAs are available to include. If you're working in a pediatric population, self-report may not be possible. You can't ask your 2-year-old to read this questionnaire and select their choices. You're obviously going to have to rely more heavily on observer-reported and clinician-reported outcomes, or possibly a perfo as well. The same can be said if you're working in a condition that has neurodegenerative manifestations, where older adults who are deteriorating with Alzheimer's may not be able to respond for themselves. So you have to look at what makes sense in your trial population for how you're going to ask these questions. And then you have to find the tools. And there are many, many tools that have been developed over the years. There's excellent 4 guidance series that the FDA has put out on how to develop those COAs and build them out to be really rigorous and scientifically sound. So you want to identify tools that meet those criteria. And sometimes in your specific population, that means you're going to use an existing COA. Sometimes it means you're going to need to develop a new COA. And sometimes it means you're going to need to modify an existing COA to make it more applicable to your patient population. And all of that work leads to you being able to say, hey, this COA is going to be fit for purpose in this clinical trial setting, and has a clear rationale. So those are really key components of COA selection, regardless of the disease. So if we want to move to the next slide, there are, however, some inherent challenges when you introduce the rare factor. Very frequently, we don't have natural history data on these diseases. So it can be really difficult to identify how we're going to measure benefit in these populations. There are very few disease-specific tools that have been developed. So you're almost always looking to either adapt an existing tool or use an existing tool in your rare populations. And there can be some challenges with doing that. The patient population tends to be very heterogeneous in rare diseases. It's not exclusive to rare disease, but it seems to be a hallmark of rare diseases is that patients present with different symptoms over time within themselves, as well as across patients. Also, by definition, it's a rare disease. We don't have a lot of patients, and so that really limits our ability to do some of the psychometric and statistical analyses with our COA tool that we want to do. There just aren't enough patients to really make that work. And then there's just so much uncertainty now with how certain treatments are likely to impact patients based on genotype and phenotypic variation that can be really challenging. And so, all of these things combined can make medical product developers hesitant to work in the rare space. I think we've seen certainly from the folks Alex and I are working with, there's a lot of activity happening here, though, and I think that's really encouraging. But there are challenges we need to tackle. So if you want to move to the next slide. So as part of the C-Path efforts here and in our collaboration with FDA, we launched a rare disease COA consortium. And the mission is to be a pre-competitive multi-stakeholder collaboration where we're aiming to identify COAs and as well as sound methodologies for collecting clinically meaningful data in rare disease patient populations. And the hope is to really make this speed the process from the bench to clinical trials and ensure that the COAs that we're including in our rare disease trials make sense and are measuring what we want them to measure and are sensitive enough to capture that change when it happens. So if you want to go to the next slide. So the Rare Disease COA Consortium, we launched in January of 2022, and we're structured ourselves to be the center of what we do is to build out what we're calling a rare disease COA resource. And I'll go into more detail on that in a minute. But we've also been very lucky and we have partners from patient advocacy groups that we've been doing outreach with, as well as trying to tackle some of those methodologic challenges that I was talking about earlier. So really making sure that we have brought together experts in COA scientist measurement, rare disease measurement, and patient advocacy, and really trying to make that pre-competitive space work for rare disease to expedite identification of the COAs and better treatment options. You want to go to the next slide. So this is our resource development process. We started with a domain prioritization and definition, and then went to a landscape analysis for the COAs that were selected. We have some criteria to whittle things down, and then we do an in-depth gap analysis of the COAs that are included to really measure them against all the evidentiary expectations that we've ever heard of from both FDA, EMA, and regulators worldwide. We have a consensus process where we have some internal expertise within our consortium membership that has helped kind of define which tools we want to move forward. And then we've also convened advisory panels to bring in outside expertise to make sure we're not missing anything. And then we're building all this information into a resource that will be publicly available in June of this year, actually. So it's been a long time coming, but we've put a lot of work in progress there. We are focused right now in pediatric non-oncologic patient populations, and the resource that we'll be launching in June is going to cover the domains for self-care, gross motor function, fine motor function, and communication and language. And the real reason for this resource development is we know COA selection in rare diseases is hard, and we want to make it easier. And so we can't create core outcome sets for each rare disease. There's too many of them. It will take forever. But we can identify tools that are really good at measuring certain domains and identify the best fit, not the best fit, but the tools that might be most applicable across multiple rare diseases. So we're not identifying one communication tool that everyone should be using. We've identified 10 communication tools, and depending on your specific context of use, you'll have to decide what you want to include in a trial program. But now you don't have to do all of the landscape analysis and the gap analyses. We've done that work already, and that's where we really see the opportunity to accelerate medical product development. You want to go to the next slide, Denisha. So as I mentioned, we are launching our rare disease COA resource in June of this year. So we're very excited about that. You'll be on the lookout for additional information coming out about that. We also recently had a paper published in Value in Health on approaches to the assessment of clinical benefit of treatments for conditions that have heterogeneous symptoms and impacts, potential applications in rare diseases. So getting back to those methodologic challenges that I had talked about previously, we did a lit review, and these are the lit review of methodologic challenges or methodologic approaches, and then linked them to FDA approved labeling claims language and to see kind of what was working, what was getting into the labels. And so that publication was just out, it was just released in April. So we're very excited about that. All right, we're going to go to the next slide. With that, it's the introduction to the Rare Disease COA Consortium. And with that, I'm going to turn it over to Alex.
Thank you, Lindsay, for this great presentation. So taking over, I want to talk about how RDCA-DAP and the RDCOA consortium that Lindsay leads work together, and how RDCA-DAP can help facilitate our efforts. So C-Path currently has a rare and orphan disease program with over 10 active efforts, and that includes several consortia for rare neurodegenerative disease, Huntington, Duchenne, sickle cell disease, polycystic kidney disease, kidney transplant, and rare ataxias, but also new pre-consortium efforts that are working very hard to potentially develop full-on consortium that includes the work for lysosomal storage disorders and one for alpha-1 antitrypsin deficiency. The RDCA-DAP program, the Rare Disease Cures Accelerator data and analytics platform, acts as a collaboratory, and we really work to make the glue between all the rare disease programs at C-Path, coordinating and collaborating with our colleagues, trying to provide help, and also providing a platform for their rare disease data aggregation. As such, we are also working with Lindsay and the RDCOA Consortium, the focus of today's presentation, by providing a place to aggregate COA data and a program to facilitate some of their activities. Next slide, please. So again, today, just want to insist on the synergy between our efforts, which might not be obvious to all. And for that, I'm going to use a case example of a work performed in Friedreich ataxia. Next slide, please. So the RDCA-DAP platform, it is a brand new platform that was launched over a year and a half ago, a place for aggregation of rare disease data from different sources, including data from industry and real-world data, but also capable of integrating multiple different data types. Each data set that we receive is shared to the platform, is going to be cleaned, standardized, and curated into the platform data model. And then we're going to make the data set discoverable to external users. Our users will be able to request some of the data that is shared to us by the contributors. If we grant them access, we are going to create them a workspace that is online, private, and secured, and we will make the data, patient-level data, available on their workspace with analytical tooling that they can use to conduct their scientific work. Next slide, please. The RDCA-DAP is not just that platform, we're also a program that works to transform data into actionable solutions. So we work on improving disease discoveries, we work to develop biomarkers and endpoints, we develop drug development tools to facilitate drug development. But we also work to advance C-Path and the RDCOA consortium work on patient evaluation tools, including COAs and PROs. Indeed, we have a database that currently contains more than 26 different rare diseases with data. And that database is very enriched in patient-level data, including outcome measures that were collected in clinical trials and natural history studies. So that really creates a wealth of information that can be leveraged to inform the work of the RDCOA consortium. Next slide, please. So it's really important to share patient-level data to RDCA-DAP because it will help accelerate the work of RDCOA. Or data, for what reasons? First, our data management team, really, when we get data, they perform a very involved curation effort to maximize the utility of the data we receive. And so we standardize the data, and that standardized data is sent back to our contributors. And we can also provide recommendations and feedback to help them enhance their future data collection and help them conduct better natural history studies that will, in return, provide better data that can feed back to the work that we do on COAs. Data in aggregation in the platform, we accumulate data, and that is going to enhance the informational and analytical value of the collected data, both because we aggregate data in the same disease, but also because we also aggregate clinical outcome measures and patient data and endpoints that may have been used to assess patients in different disorders. Finally, we have a data analysis and a quantitative modeling team, and the work that we do can help with the COA work. First, because it's going to help to inform COA selection. We can help optimize COA interpretation. We can inform new COA development. And as I mentioned before, we can also help help our contributors to improve natural history studies conduct. Next slide, please. So to give you an example of some of the work that we do that can help inform COA work is a case example in Friedreich ataxia from the FAICD for Friedreich ataxia integrated clinical database. This is a collaborative effort that RDCA-DAP is doing in partnership with the Friedreich Ataxia Research Alliance. Owing to the effort of FERA, we really created the biggest database in Friedreich ataxia in the world right now, with currently 6 clinical trials, a big natural history study from FERA, and that database is continuing to expand. We just recently got data transferred from another natural history performed by EFACT, the European Friedreich Ataxia Foundation, that recently joined our group. And we have more data in Friedreich ataxia that is going to continue to increase that very big database. The analysis that I'm going to show really today were conducted on a subset of patients containing over 1,300 subjects and more than 5,000 collections and observations on patients. So to get a sense of the amount of data that we have in that database, the bottom left graph shows you the number of patients per study over time, just give you an idea on how many patients that we have available for each study with collected data. One of the key outcome measures to track Friedreich ataxia patient progression is a scale called the Modified Friedreich Ataxia Rating Scales, or mFARS. This is displayed on the low right figure, where you can see with the connected black dots, these are patients' individual trajectories on the Friedreich ataxia rating scales over time throughout the database. And in red, this is the average to show you the average patient population progression on the scale over time in the FAICD cohort. The next 2 slides I'm going to show you, next slide, please. These next 2 slides are really some of the work that was conducted by FERA scientist Christian Remy. The first slide shows you an analysis of the MFARS variability at baseline. Here, the baseline is really when the patients enter the different trials or the natural history study, meaning that they may very well have already started their disease, and it could be a picture of different, it's a picture of different patients at different stages of their disease. So the mFARS is a multi-composite scale with 18 different patient measures or items that are spread across 4 domains, A, B, C, and E. So what you see on the left axis, it's the proportion of individuals in the data sets. And we bin these patients by severity from minimum to maximum on each unit of the scale. So from light red to stronger red, the patients are going to score more and more when the color gets darker on each items of the scale. So really, this work shows you the variability at baseline, but it's very informative, because what you can see when you take a look at the E domain is that a lot of patients are already scoring very high on these items, and some are even maxing out. And this is when really the patients enter the studies. So that gives you some insight that potentially that domain might not have been the best suited for that particular cohort of patients. And you can imagine that such analysis is going to lead plenty of additional insights to help you select the best collections and measures, especially when you're at the stage of wanting to conduct a new study. This is tons of information that is valuable to help you decide what data you want to collect on the patients. The next slide really shows you another example of patient variability on that scale. So here, this is the total mFARS score, and we're showing the longitudinal variability with respect to the rate of progression. So on the left, this is the total score. And then here, the patients are bin by age of onset. And you can see here that when patients start the disease later in age, in purple, the progression is much slower on the scale, while when you take a look at patients that start the disease at a very early age, they are scoring faster on the scale and seem to be maxing out faster than other categories of patients. So again, what you see here is an illustration that on how, taking a very deep dive in an aggregated database, you can extract valuable information that tells you how valuable the scale that you use in these patients was, but also helps you and guides you on potential adaptation you would want to make. For example, here, we may need to, that figures may lead us to think we may need to rethink how to better track the patients that start the disease early in age. Next slide, please. So in conclusion to this presentation, as Lindsay conveyed, we have that question, does one size fit all? Well, one COA may be applicable across multiple rare disease, but the assessment of a trial-specific context of use is required so that we establish the proper COA as fit for purpose for a particular study. I also want to re-emphasize that data sharing is extremely important to inform COA work for interpretation of COA selections and development of new COAs. In the future, in partnership between RDCA-DAP and the RDCOA Consortium, we are going to continue the work. We have some early plans of evaluating some of the tools that are used to assess patients across multiple disease areas, potentially, just for an example, the Vineland, a tool that is classically used in neurodevelopmental disorder. That could also be GATE, as we have many different rare diseases in the database with the GATE components. We also have early stage of development of new applications for COA that are going to leverage the platform technology, in particular the ontologies that our team is developing. And finally, we are going to continue to work to optimize data sharing specifically to inform the COA work that Lindsay is doing. So that ends with the presentation, and I'd like to welcome our three panelists today. Jennifer Farmer, Chief Executive Officer at the Friedrich Ataxia Research Alliance, Cheryl Pease, Director of Patient-Reported Outcomes at Janssen, and Michelle Campbell, Associate Director, Office of Neuroscience at FDA. And welcome back to Lindsay for this panel. Before we go in the Q&A, we have a great attendance today, so I'm hoping, and we have plenty of time to address all of your questions. So please continue to input your question as we discuss, but maybe let's just do a little wrap up and kick off with our panelists to get some of your comments. Jen, if you want to start and let us know a bit more about, where do you see the value in the efforts that we're doing, and just, give us your input on how informational that this kind of efforts can be for you as a foundation leading major data collections on patients.
Alex, Lindsay, thank you for inviting me to participate today. Maybe just a little bit of background. Alex, you did a nice job of talking about the FAICD, the database that we've collaborated on building for the Friedreich's ataxia space. But for folks who don't know, our FARA's process here started 20 years ago. So we started a natural history study 20 years ago. We started thinking about clinical outcome measures even a little bit before that. And Lindsay, I at the beginning when you were talking about some of the background about COAs, we've done them all, right? We borrowed existing COAs, we developed our own COA, and we've modified COAs. And it is because in a disease like Friedreich's ataxia, there are different symptoms and different contexts of use that we're trying to cover. And there are still things that we can't assess really well, where we're continuing to try to look to develop new COAs. And so I think just in terms of, this is an ongoing process and it's not a one and done. And I think that's an important mindset, especially from a patient advocacy perspective, that has been an important learning for us. And I don't think we talk about that enough, but this is, it hasn't stopped us from going forward and doing the research, but it is an iterative process, and you have to continue to iterate it. And it's an important mindset and an important realization. But these, Alex talked about this data set that we now have. It's our natural history data that has a lot of data on these clinical outcome measures and these clinical trials that have been done in the past in FA. And it was about 5 to 7 years ago that enough of these trials had been done, and several of them had had negative outcomes. And we realized that there might be some differences, right, in how these COAs perform when you're collecting them in natural history versus when you're collecting them in clinical trials. And one of the really powerful things that's now coming out of this combined data set is we can understand that, we can interrogate that. So we can understand the placebo effect that occurs in our disease that we, learn about the hard way through doing clinical trials and then seeing it, but having not planned for it well enough in advance. And realizing that as we, the next generation of clinical trials, we can learn from that and build on that. So, this aggregated data set is just, it's absolutely necessary for us to develop that next generation of clinical trials better and smarter. And so we can do things like placebo effect. The other thing we're looking at right now is, because we have all of this data, does it give us other ways to design trials? Can we use a Bayesian borrowing approach? Do we have enough data from other trials that have been done or natural history that we can supplement placebo arms of some of our future trials with some of this data? That's, an experiment, data we're looking at right now to see if that's a feasibility. So, it's, I guess I'll just end by saying, the possibilities continue to grow when you bring all of this data together. And I'm excited to also, see if, as these data sets come together and some of these COAs are used across different rare diseases, can we accelerate our ability to develop them in any one disease, because we now have the ability to kind of look across diseases. And, I've shared some of the COAs that we've developed with other rare disease groups, but we haven't had this ability to share data across a platform like this and look and see how these measures function differently across our different diseases, and how does that help us iterate better in the future.
Thanks, that was great. Well, I'll give the mic to you now, Cheryl, and after what Jen said, can you touch point on your perspective, especially coming from industry? How do you strategize the selection of COA before engaging in a clinical trial? How do you see these efforts could be helpful for you? And just mostly give us your perspective.
Yeah, Jen, that was really well put, and I totally agree with everything you said. First of all, I want to thank you for the opportunity to participate as a panelist in this webinar. I think it's really great, and personally, I enjoyed hearing about the example from Friedreich's ataxia and the commitment to advance the science in rare disease by promoting sharing of existing data. There are a few points that I'd like to make to communicate related to the COA selection in rare disease. Rare disease are complex conditions that have debilitating impact on patients, which we know. Their lives are, so the evaluation of these impacts are critical in our research. As we think about selection of appropriate COAs, we must look at all the available data resources. The database could be a useful resource in our toolbox that is data-driven and constantly being updated with information. I also wanted to highlight the point that partnerships with patient advocacy organizations are paramount in today's climate as well. We always hear about, rare diseases, difficult rare diseases, complex, but it's so rewarding. And one of my favorite parts is being able to hear from the patients and working with the patients. So that's just an important point that I wanted to make from my perspective as well. Oftentimes there are not disease-specific measures already available, so we're left with selecting instruments based on research of available information. Availability of data that provides insight into how the instrument's going to perform is going to be really helpful. This is something that could be really exciting to have a glimpse into how the measures perform as well. It could provide answers to whether the instrument will be sensitive enough to measure change in the clinical trial setting. And this is especially useful in the situation where we have shorter timelines and minimal opportunity to pilot the COAs. I'm looking forward to seeing how the resource is utilized and implemented in rare disease research.
Thanks, Cheryl. Lindsay or Michelle, does it trigger any reaction from your side?
Thank you, Alex and Lindsay, and thanks to Cheryl and Jen for sharing their thoughts and experiences from the groups they represent. I really find the combination and presentation of today's efforts of both the Rare Disease COA Consortium and RDCA-DAP a true synergistic opportunity for us who spend most of our times, and if we are in the measurement and COA world, how can we leverage something such as data sharing could help inform us and ultimately in our selection of a COA to support a clinical trial endpoint. All of us who deal with rare disease drug development face the same challenges every day. We talk about it almost every day amongst ourselves, and definitely when we have our monthly meetings with the COA Consortium. But I think this offers such a great opportunity for us to think about how can we data share not only results from clinical trials, but is there that opportunity for the all-encompassing qualitative data, often our foundational information from talking from patients and families and their caregivers about their lived experience with their disease and disorder. So I really do think this is a great opportunity for us to think about for future collaborations and opportunities that may be able to help inform drug development. And we know that our sample sizes are so small, and we really need to make sure we have the best information possible to develop a trial design that will provide us with the most interpretable data in the long run. So I'm really excited that we are having this discussion today. I know this will probably lead to many more discussions, but I want to say that I think this is something that we really need to be thinking about as we continue to advance and accelerate advancements in rare disease drug development. We are all stakeholders together, and I do think that this is one opportunity that may be a really great time for us in the measurement side to really see how data sharing could help us. I'm also excited with the upcoming launch of the COA resource that I think is going to be an informative starting point for folks to begin their conversations when they're thinking about how they would like to assess concepts in their rare disease clinical trials. So Alex, I'll turn it back to you.
Thank you. I think unless you want to add a comment, Lindsay, we can start with the questions from our attendees. Sure.
So I see there's a question here. How's the Rare Disease COA Consortium handling qualitative data? So I'll be perfectly honest, at this point, we are not. We've been doing a lot of literature reviews on existing tools, but have not reached the point yet where we go out and collect additional qualitative work for any given disease or cluster of diseases. I do very much think that that's somewhere we could go in the future. The resource that we're developing is limited to the tools that are available now and have published data around them that we can evaluate. But we know there are tools being developed and being used in rare disease clinical trials that just haven't been published yet. And we're keeping on top of that as much as we can. And I also foresee that there will be domains that we look into where communication is one that comes to mind. We identified a number of gaps where there just weren't good tools to measure certain aspects of communication that I think are still of interest to the rare disease community, and where we may need to go back out and do some additional qualitative work to fill some of those gaps. But I do think, and Alex, you can correct me if I'm wrong, but qualitative data can be submitted to the RDCA-DAP. So if
yes.
you've got data, if you've conducted qualitative interviews, and I think one of the things that I always think about is, sometimes programs fizzle out while you're doing COA development, and those transcripts that you did your initial content, validity or concept elicitation interviews with patients, they just kind of fizzle out and go nowhere. Think about submitting them to the RDCA-DAP, because somebody else might be able to pick it up and use it later. And that's really making sure that we take advantage of the data that the patients have given to us and maximize its use. All right, so the next question is, Alice, this one's going to be for you. How do you partner with rare disease patient groups that have registries in place? And is there any support for these organizations to keep their registries running?
So good question. So just to be perfectly clear, RDCA-DAP doesn't collect data. So the program and the platform was really created to be a place where people could share data in a very secured manner with a lot of flexibility in how they want to share data to the platform and open their data to rare disease communities. So we don't engage in data collection, and we're not a registry. What we're able to provide is, so we do a lot of outreaching, communications. We have a lot of data from registries and foundations. And when we discuss and we finally reach an agreement and someone shares data to us, we're able to provide them with the feedback on their data collection. And I'm hoping actually to have a webinar with the Rare Epilepsy Foundation later this year, where there were really early adopters of the platform, share their data. We did that work of curation, provided them with a recommendation, and they made some changes to their collection and updated their data. And we're going to get a new data set of their improved collection. So this is like an example of how we directly help with the registries. When it pertains to putting a registry in place, this is not our work. We have an ongoing collaboration with the National Organization for Rare Disorders. So, I would refer you to discussing with Nord if you don't have a registry yet and you intend to build that.
I got a gentle nudge on the qualitative question as well. We actually, the Rare Disease COA Consortium does have a qualitative working or subcommittee working right now, where we're looking at how to apply qualitative data collection in rare disease trials. We haven't collected any qualitative data, but we are looking at how you can leverage that in your trials.
There's a point that maybe we haven't discussed, and I think it would be great to have Jens and Sherry's comment on, is how helpful it is when you're the effort is to inform trial design. We do have active industry users of the database, including the Friedreich ataxia database, but also other rare disease effort and some example of industry that worked and developed some work to help them with their clinical trial activities. We also have some examples of a previous industry that work in a specific rare disease area, made a change in how they were planning to conduct their trial and changed, the outcome measure by learning, joining our teams and learning, new things on particular measures. So maybe if you can comment on that, Cheryl, maybe if you want to start.
Yeah, I can start. So I would say trial design is very much a part of COA selection. So, some trial study designs are not optimal for, collection of certain types of COAs. So when I'm putting together a PRO strategy or a COA strategy, the study design is very much part of that. And if we have information on how a measure will perform or a glimpse into that, then it could influence also like the administration schedule, when is the best time to, kind of collect that information, and even further to that, the optimal endpoint for that particular measure to feed into.
Thank you. Lindsay, maybe a question for you. They say the example of using existing database to inform COA across different COA items is helpful. In your experience, do you have an example of selecting COA measurement for the same item? For example, different PRO measurement scale exists for the COI of functioning.
Right. So I think if I'm understanding this correctly, is how do you decide kind of ultimately which tool you want to select? Because there are frequently multiple COAs that measure the same concept of interest. So things like physical functioning or self-care. So that really gets back to going back to the roadmap and understanding your disease population and understanding what you think your treatment is going to impact, to identify really which tool might be the best. Pilot data might be really helpful as well to really understand if you've got multiple tools that might work to measure that concept of interest. How do they align with how patients and caregivers are talking about their disease in terms of their content? Do they have floor and ceiling effects that are really obvious from the get-go? Because you can throw a tool in, but if everyone's already on the floor, you're not going to see any improvement because everyone's already, off the scale. So you want to make sure that you're picking tools that there's room for your patients to improve or stabilize. So you have to make those kinds of considerations when you're selecting the final tool that you want to use.
And maybe a related question. Do you believe that PRO tools used across multiple rare disease may actually replace disease-specific tools in clinical trials, or rather it is that they will be used in addition to disease-specific tools that will still be required for future regulatory submissions?
Oh, I wish I had a magic 8-ball for that one.
So I can weigh in for a little too, if you want. My reaction to that is, the generalized tools are probably better for more distal concepts where, disease-specific tools are many times or oftentimes would get at those proximal concepts. So I think that one, it depends on the disease and what you're looking at. Two, it depends on the messages you want to generate and what messages are appropriate for different stakeholders. So like we would be targeting different things if we were talking about FDA or HTAs or physicians or patients. So
Yeah, and
go ahead, Michelle.
I could chime in briefly on that. I think I think number one, it's a hard question right now, as rare diseases, are so heterogeneous, and so I'm not sure we're at this more broader disease specific concept in the individual, disease, the bigger concept. I do think though that as we explore and continue to learn about maybe families of diseases that may have some similar features, they may help us be able to incorporate a couple broader type COAs and maybe in all of their trials. But I still think in the end, and I think where Cheryl was going with her response too, is that in the end, a lot of it will be driving by what do we think our treatment can actually intervene and do. And so we need to be able to measure that. And so I still think that we will be having those. But, we don't know as well as where science will take us in a decade or so. And maybe there will be a shift. But I think for the time being, we're still, the need is still there for those type of disease specifics. But I think that doesn't preclude the idea of if we have opportunities to put some broader things, maybe, like I said, in a family disease concept that could help inform, natural history or future development aspects in various diseases.
And Michelle, we just actually had a question, an FDA question. Given the heterogeneity of rare disease, would FDA be open to combining several established functional outcome measures as a primary endpoint in a clinical trial? For example, the North Star and PUL, I don't know this one, to capture both upper and lower mobility.
Yeah, the PUL is the test that's often used in our Duchenne trials. So we recognize obviously that heterogeneity is in all of our diseases, and our patients and are expressing who are participating in trials may have different aspects of their disease that is their symptoms that they express. So I think in that term, I think number one, I think those are conversations that an individual drug sponsor needs to have with their appropriate therapeutic review division. And again, it has to kind of understand what do I think my drug is going to improve, right? What are we trying to go for in the end? And I keep on mentioning that because sometimes we really do have to find this balance between what's important to patients, what I want to measure, and what is the drug supposed to do, or the medical product treatment, is supposed to do. I think when we look at our functional measures, where we want to take different things, I think ultimately determining what a hierarchy is going to be, those are those conversations that you definitely want to have early with that therapeutic review division, and what will really make sense, what will still allow good interpretation of the data. Sometimes when we try to combine things, we actually lose the ability to interpret if there is change that may occur. And so we obviously do not want to lose interpretation, particularly if we if a decision has to be made whether or not to continue on a trial, and because we accidentally combine things, we lose that interpretation ability, and we may make a choice that was not correct because of that COA choice by combining. So I think that's a great question. I think that's a question that should really come in to be asked, depending on that program and what makes sense, and how does it, I think a lot of things align. How frequently are these things being measured? Do we expect change on these things? All of those things have to be taken into consideration. So do come in early to have those conversations with us to really think about why this is the approach you want to take, and does it really make sense?
Thank you, Michelle. And actually, you mentioned, patients, and maybe Jen and or Lindsay, if you want to comment on that question. From Christine, how often do you listen to patients' inputs with regards to commonalities in their disease when forming trials?
So, Alex, are you asking how we incorporate sort of the patient feedback into the design? Was that the?
I think so.
That's part of the question. But there's also, I think, a point on the frequency, because it's explicitly the question is, how often do you do that?
Yeah. Well, when we try and do that all the time, right? As a patient advocacy group, that's our job, right? So we try and do that all the time. But, we early on in the development of the natural history study and thinking about clinical trial outcome assessments, what we could use, what tools could be available. It was very much understanding the patient perspective of the disease. So we brought together the patient community, the clinician community. What can we actually measure? And there's a dialogue that occurs that, we make sure we're capturing what is clinically relevant to people, but doing it in a way that's reliable and robust as best as possible. And, that's what we try and facilitate. But, how often do you do it? That's a great question. So there was just Friedreich's ataxia. We just had our first approved treatment. And we're I can see what's gonna happen, right? Because we now have an approved treatment, and we need to keep collecting now the data, because we need to know, are these same tools going to be relevant for future clinical trials? Or does our interpretation of these tools change in the context of an approved treatment? We don't know the answer to that yet. So, I think you have to continue to engage in this process. It's not a one and done. And it will change depending on the symptoms you're trying to address. And as I think Michelle pointed out really nicely, that even for Friedreich's ataxia, there are multiple different treatment approaches, right, that we're trying to advance, or that our industry partners are trying to advance. And so the COA for one trial might not be the same for another trial because of the drug that's being tested, that they have to be different. So that was a little bit of a convoluted answer. I apologize, but I think I was trying to communicate a few different points. But I think the main one is that yeah, you have to continue to engage the patient community in understanding the symptoms continuously, and especially as treatments evolve and your understanding of the disease evolves.
Thank you. No, that was great. We actually had a question for you on, what's next now after this very, very important milestones for patients.
Yeah, I think we all we all look to the day when our work is done, right?
Yeah.
At least when I started, I was like, oh, we'll get a treatment and we'll be done. We're out of business. And I'm still working towards that. But yeah, we have to continue to collect this data, the same data we've been collecting in the context of a new treatment. We have to continue to make it available, because now drug developers want to know, how do we have to change our clinical trial design if patients are taking now this approved therapy? How does that change our future design? And the only way to answer those questions and facilitate that is continuing to do what we've been doing, I think.
Thank you. Cheryl, I think is that the last question you want to, or Michelle, that you want to address?
Sure, we can ask that last question if you want to read it, Alex.
Well, I think it just disappeared from my side.
I think it was a question about individualized endpoints.
Yes.
So it's a great question, and something that we see be asked a lot when we are providing advice in individual drug development programs. I would actually refer folks to not only the multiple endpoint guidance about considerations when selecting endpoints, but actually the patient-focused drug development guidance series, particularly guidance 4, which just actually came out 2-3 weeks ago. Excuse me. That actually talks about how do we really get to that endpoint when we have our COA and we've collected all this patient data. It's the ultimate guidance that we've been waiting for. They really talk about how do we get that. And they do talk about the challenges in some of this approaches. And I do think that the article that Lindsay and the Rare Disease COA Consortium actually kind of hints at this a little bit, is that it does talk about the challenges and when we try to do this approach. And so I would encourage folks to read that. I think this is the conversation you want to have with your review division very early. A really important thing to understand is that natural history and how we think the disease will progress. And how long is your trial? So there's a lot of nuanced factors into that. I would also encourage folks that on May 4th, there will be a webinar on the guidance 4, which is on how do we go from a COA to an endpoint. So the FDA is hosting that. I believe it is at 1 or 2 o'clock. I should really know, shouldn't I? I'm so sorry. I believe it's at 1 o'clock. I think that could be also very informative. That is a very common question we get. We understand why folks may want to take that approach. It just has to make sure everything is well thought out, and there's a lot of nuanced aspects to that. And I don't know if Cheryl has any thoughts, because I'm sure this is a question that she's also faced sometimes as trying to put together that trial design. I don't know if you have any last minute thoughts on that.
Yeah, no, definitely. I just, it's get to know your patients and get to know what their biggest unmet need is. What are they hope for in, getting symptom relief? It's, getting to know the patient. And then as far as endpoints go, then you go from there and say, okay, how do we measure this? How do we make this into an endpoint? And is it possible with the study design that we have set up? So it's kind of a big collaboration of, thinking through, putting a lot of thought into what you're doing and having a good, foundational reasoning and support behind it.
Thank you. And I think we're really close to the end. So I will take the last question. But first, thanks everyone for joining. Thanks to our panelists. That was really great. If you have any follow-up questions, you can reach out to me or Lindsay, and the webinar will be made available online. There was a very last question from a foundation on how do RDCA-DAP handles patients that potentially were incorporated in multiple data collections effort. Well, that's a question we can discuss during our next webinar, which will be on May 18th, about GUIDs and de-identification tools available to the rare disease communities, so that you can prepare for tracking patients in multiple collections over time, but we will talk about more broadly, on how the identification is really important, not only to protect patient privacy, but also for prospective linking of future data collections. So thanks everybody for joining today, and we'll see you again on May 18th.